Hi, my name is Rebecca Burdine, and I'm an associate professor at Princeton University. I'm also mom to Sophie, who has the deletion form of Angelman syndrome. Today, I'd like to introduce you to a field of research that will be critical for helping us understand how Angelman syndrome affects neurons and for helping us develop therapeutics. Researchers Eric Levine and Stormy Chamberlain, both at the University of Connecticut Health Center, are working with human-induced pluripotent stem cells. You may have heard these referred to in the news as IPS or iPSCs. Dr. Levine will tell you about his particular project in just a moment, but I wanted to tell you why I think this research approach is so powerful. Using special techniques, skin cells from an individual with Angelman syndrome can be coaxed or induced to become a cell type that can give rise to any other cell type in the body. They're called pluripotent because they have multiple choices as to what type of cell they could ultimately become. In our case, they'll be given signals to turn them into neurons, and these neurons can be studied outside of the brain in a dish. Also important, these neurons will imprint, which means they will silence the paternal UBE3A, and since they come from an individual who lacks UBE3A due to Angelman syndrome, these neurons will have Angelman syndrome and can be studied in a dish to see how UBE3A loss affects functions that neurons have, including communication and growth. It will be important to understand these deficiencies in neurons if we're to develop therapeutics to target these types of issues. And while we have really important animal models for studying Angelman syndrome, it's important also to have this human neuron cell type in order to test drugs and therapeutic approaches and make sure that they behave correctly in a human cell. Now, before you hear more from Dr. Levine, I just want to take a moment to thank all the individuals with Angelman syndrome who donated skin cells to make these studies possible, and the Angelman Syndrome Foundation for supporting such important work.
My name is Eric Levine, and I am a professor of neuroscience at the University of Connecticut School of Medicine. I would like to tell you about my research project that has recently been funded by the Angelman Syndrome Foundation. Imagine a traffic light at a busy intersection. It directs the vehicles so they flow properly across the intersections. Similarly, in the brain, there are traffic lights that control the intersections, also known as synapses, between brain neurons. In an individual with Angelman syndrome, we believe that the flow of information at certain synapses is not regulated properly, and this could lead to symptoms such as seizures, developmental delay, and movement disorders. In my laboratory, we will use a recently discovered technique that allows skin or blood cells from an individual to be converted into brain neurons that can then be studied in the laboratory. This will allow us to study how the intersections or synapses of human Angelman syndrome neurons are working, which is a first for Angelman syndrome research. Specifically, we will be looking at the process that adjusts the strengths of these connections or intersections. This process, known as synaptic plasticity, plays a critical role in the formation of circuits during brain development and in learning and memory throughout life. Malfunctions in this process could be responsible for language impairments, motor disorders, and seizures, as well as other aspects of Angelman syndrome. Overall, this research will do two things: identify the role of this signaling process in human Angelman syndrome neurons, and provide a platform for discovering new therapeutics and for screening these drugs in human Angelman syndrome neurons. We're excited about the potential benefits of this research for individuals with Angelman syndrome, and we greatly appreciate the support of the Angelman Syndrome Foundation in making this research possible.